research versus exploration.
Yeah, it's
In
self-to-cliff.
Dr. Ballard's book,
So
Into the Deep,
it's
I'm just about
actually perfect
halfway into
to
it
the
now.
20-meter range,
He talks
I
about
guess.
early on in his career, he kind of encountered this stigma that exploration and going out to sea with no,
you can see, so his heading is basically 270
hypothesis ahead of time or set plan, so to speak, to find things. It wasn't quote unquote real science or real research. Did you ever encounter any of that like sentiment early in your career?
Yeah.
Or is that kind of?
I have encountered it.
Yeah.
You can do 20-meter.
But not in the places you would think.
Okay.
You know, I think there has been a greater emphasis in recent years, maybe the last 10 years or so, on exploration, in part due to Dr. Ballard's efforts. You know, now we have the Okeanos Explorer, a dedicated ship of exploration, as well as other entities like OET among them.
Right.
But I have encountered that at the academic level, you know, in the university. But you know, I think that there is value to a lot of the things that exploration science can add to you know, our understanding of natural history. You know,
overherc, it won't matter.
It's hard to go out to collect and understand, you know, the natural biodiversity of the environment if really we're only, you know, targeting hypothesis, you know, driven research questions, because oftentimes if you're granted funding by a agency or some entity to answer a specific question, you want to go to a site in the ocean where you know you can find the material you need to answer that question.
Right.
Whereas,
bring your head to the left just a little for me.
Swinging over right now.
You're going to go to some place that's been visited before, most likely. Some place you know you can get your samples. But you know, if you go to the same places all the time, you never really understand anything new about
it won't come around
you know, spatial patterns or temporal patterns in biodiversity.
Right. Right.
So like that, you know, that's my take on that question. So, you know, the collections we make here are going to be totally different than, you know, perhaps, you know, a frequently visited site that is known to have, you know, high density corals or something like that.
Gotcha. Thank you.
Yeah.
Would you say the majority of the scientific community today, at least in regards to biology, would view exploration as a good means of conducting?
I think it's still poorly understood, and I think that gives value to what we're doing out here. Yeah.
Yeah.
You know, the goal that I have always held, the objective that I always held, is that exploration should come first to help formulate, you know, hypotheses that can be investigated later. And I think that's also now being supported by some funding bodies, which is positive, positive direction. Yeah.
So you're saying it kind of lends itself to being the first step of the scientific method almost.
Yeah, yeah,
it's just
you could say that.
A small one, I think it's going to...
Thank you.
Yeah.
Looks like it's going to go around the corner. It's hard to say. You can't think ahead more than 20 meters.
A lot of bases up here that are broken.
Answering that question in chat, who's sitting on the other side of the window behind Steve? That is our ship-to-shore interaction studio. So we'll reach out to schools or other types of programs that are interested in oceanography.
Yeah,
and then we make calls to them, and we have a face-to-face interaction.
Is there any rocks that are loose? Loosey-goosey? We can try and break some.
Loosey-goosey
rocks.
rocks.
I can think of that. A lot of, I believe these are precious coral bases, the small calcium carbonate discs on the rock, but there are some sponge ones too.
Do you want to try to break something off, Dan?
You want a closer look at them, Steve? Or?
Yeah, we can. What about this right here?
Yeah.
Yeah, can we try and pick up this rock? Oh, under there.
Sure. Nah, I think we're 20 meters is all right. What rock are you after?
Yeah, it's, can we try and either pick this up or break it off? It looks like you get it. So your fingers around it.
Maybe.
Maybe that or something. It's tough. It's this a lot of crust in this area.
Delta's at 1.8. Are you happy there, or do you want to come down a bit?
You can come down 5 meters. You're well off the cliff now, but we're gonna just keep an eye on your 20-meter range.
Roger.
I've been known to go single digits up against the cliff. What happened there?
Here's another question from chat. Does funding for the OET operations come exclusively from government agencies?
Looks like our tether's got another little tie in it.
I don't know if I have all the information to answer that question. I know that as a part of the OECI, the Ocean Exploration Cooperative Institute, a substantial amount of the funding for this cruise comes from that entity, which is from NOAA Ocean Exploration, which is a government entity. So this cruise is at least funded in part or in whole by that process. In addition, they funded, the OECI funded our cruise at Kingman and Palmyra earlier in the year. NOAA OE and the OECI funded in part our cruise at exploration cruise at Kingman Reef and Palmyra Atoll back in March. They have a really cool mission, the OECI. If you get a chance to find out some more information about them, it's a
nice hold on that one. Looks like a climbing grip.
Highly encouraged.
Sure does.
The one we were going for was the one right behind it.
Oh,
actually, I think it has a little bit better purchase, maybe.
This guy here?
Yeah, right there. Yeah, that one.
No, needs to be more of a nub. Like,
yeah.
It's too short of a nub.
Do you want to try this guy? It looks like it might be in the sediment.
No, it's just a nub sticking out of the sediment. If you want to break one off, it's got to be a...
Okay, to keep moving and
yeah, we can keep moving.
And yeah, we'll keep an eye out for something that's either breakable or loose.
Thanks for trying.
Not done yet.
Then to answer that question in the chat, do you ever reverse the charges for those calls? I'm not entirely sure how the ship-to-shore interaction process works. You'd have to ask Megan that. But if you are interested in trying to find trying to get a ship-to-shore interaction with the Nautilus, you can go to our website at nautiluslive.org, and then you can go to the education tab. You scroll up and there's the education tab, and if you scroll down just a little bit, you have the live ship-to-shore interactions, and then you can click the request a connection And then we'll hopefully can give you a live Q&A right here on the Nautilus from our lovely crew.
Come down 5 meters.
Coming down a few meters.
5 meters.
Coming down 5.
This is pretty neat. You might find some loose rocks at the bottom of this, but I don't know if they would
You
be
see that little loop there?
Yeah, I see it.
basalts. I'm guessing not.
Yeah, probably not, but I don't know if we're going to find basalts at all for the rest of what we're doing.
Some of this stuff. I mean, that's kind of under the overhang, but
Nice.
still, that's got to be loose if you're interested in rocks.
Yeah,
We're gonna.
if you can fit down there.
Oh, you can fit down there. But this is like, I don't know, at least 60-meter drop here. So
Yeah.
but there may be little shelves along the way. On the way down, you might find some loose rocks.
Want me to keep coming down with you?
Yep.
Okay.
Oh, this one looks kind of nice, but it's pretty deep in there.
You see a rock back there that you like?
Yeah, I like this guy, but it's
I like
deep.
rocks.
It's the deepest one, so
Of course it is.
it's like
It's probably attached
Okay.
just in the sediment. but
yeah,
there's
maybe
only one
maybe
way to-
this right here.
Only one way to find out. Tilt your camera up a little bit.
Camera up.
That
Yes,
kinda
I know.
looks breakable too.
Move the boat away from the wall. You'll have to come up a little bit there.
Coming up a little bit.
Overhanging cliff. Not too, yeah, go ahead,
What
2-0.
is what?
Is that the crinoid? Yeah. Swimming.
The swimming crinoids.
Wow.
It'd be trippy if you didn't know what a crinoid was.
Yeah, it took me a second to actually realize what it was. I thought aliens were real there for a second, and now my hopes and dreams are shattered. So,
Sorry, we're a little close to the promenade there with Atlantis, so
Okay.
we're gonna have to move on. So
This stuff
overhanging
is-
cliff there.
This stuff is farther out. I don't know if what your plan is, but
We're gonna move the boat away, then I can probably come back down.
gotcha.
while we do that, someone in the chat wants to know, can everyone
can come up a
introduce
little bit.
themselves? We can do that as well, but if you're
Copy
really,
that.
really interested in to find out more
Come
about
up.
the people on watch, you can go to our website of nautiluslive.org, scroll down, and then you can see who's exactly on watch. But sure, I'm Matt. I'm the honorary science communication fellow for the 4 to 8 watch.
That's all you got?
That's, well, I mean, my
Alright,
favorite
alright,
color
no
is
problem.
orange.
That's
I don't know.
That's
Oh, really? Me too.
Hey! Look at that. Great minds think alike.
So are you happy there? You want me to go higher?
Go on, Brandon. Introduce yourself.
Right.
Happy there.
I'm
Happy there.
Brandon Scott. I'm a scientist focusing on geology. I'm a master's student at UNLV in Las Vegas. Yeah,
What's your
favorite color is orange.
Okay, great. Steve?
Can I get a glory shot of her here at the
My name
cliff?
is Steve Oskovich. I'm the biology science lead for this cruise, as well as the watch lead for the 4 to 8. In addition, I'm a postdoctoral researcher at Boston University, where I study deepwater coral ecology, biogeography, among other things. And my favorite color is precious coral.
You know what? Sure. Why not?
That is actually a real color.
Yeah,
Really?
coral is,
Yeah.
right? It's
Precious,
like an
precious
orangish
coral.
pink.
Precious coral is actually a real color too.
Okay.
It's a, yeah.
Bring your head.
What do you
But yeah,
imagine?
coral,
A little left.
live coral is a real color too, yeah.
I imagine like orangish pink.
It was apparently the color of the year a few years ago. Live coral.
Huh.
Is it
Color
saying-
of the year, that's the thing.
Yeah, that's the thing.
Kukui.
These rocks look breakable. If,
I think it's 2020? 2019.
They do.
Living
Break-
coral.
Throw
Color
my
of
hat
the
in
year.
here, Matt.
Do you want cross?
Ooh,
I lead in the
nice.
front row.
Very nice,
saying I'm
soothing,
Dan.
almost salmon color.
Yeah, video engineer for
this,
the 4 to
this
8
one
watch.
looks nice.
Roger.
And if you go on the Nautilus Live Instagram today, I'll also
Bring
be
your
doing
head to the
the
left a little
story takeover. So
Copy
you can
that.
see some inside highlights.
Yeah,
from the
aloha mai
front seat over
kako.
here.
My name is Kukui. I go to UA Chilo, and I am the data logger for this watch. And my favorite color would be Victor-Gorgia purple. I don't know if that's a real color,
that is
but
a great color.
That's a nice color.
That is such a unique color too.
Does our favorite color have to be a coral color? Can it be-
Apparently.
My favorite color is plaid.
You could say like no color sponge or something.
There you go.
Ooh,
They're all
yeah.
translucent.
My favorite color is glass sponge.
You know what's a
we,
really cool
are
color
we
too
picking
that
up a rock
corals
here or
produce is that lime green color on some bamboo corals, lime yellow.
Steve, are we picking up a rock here?
Yeah, we're gonna pick up a rock. I'm gonna let Brandon pick it out here. That one's a nice one in the back there, just above the lasers.
I was thinking
No, different one.
down to the bottom left, down here. Is it this guy? Or no, this guy. Yeah.
Good choice.
Yeah.
Grab one of those rocks.
We're gonna go in the starboard box and for these.
Can you give me a circle again, Brandon?
Sure.
Okay.
Move over a little bit.
Oh, there's a, is that an anemone?
Mushroom coral anthomastus.
Oh.
Yep.
Just gonna let the dust settle for a second. Okay, one more time. Give me that circle. Right there in the middle.
I can't see.
I think it was down in here somewhere.
Okay. All right. There's my claw.
Let's see if we can find it again.
Okay, we're out of here.
All right. Want me to stow?
No, just hold it down. Bring it down so it's in the bottom right part of the camera. Tuck it in a little. See how it's sticking way out there
Roger.
in Atlanta.
How's that? Better?
Yeah, tuck it right in. Don't
Okay.
want it sticking out here while we're close to the cliff. Don't stab
1760
yourself.
meters.
Blue button off, or are we waiting?
We'll find another rock.
Roger that.
come down on the winch now.
Coming down 5.
Yeah, good. Follow me down there.
Yep, following you down.
So we are on the search for a rock, but we're working in a really steep wall right now. So we're just trying to maneuver into place.
If you'd like to know how steep, you can just pop on over to channel 2, and you can see Atalanta's point of view of just how sheer this kind of rock face is.
Oh, wait.
Tilt down a little with your camera for me. That's down far enough.
Tilting down.
Tilt down a bit more.
Coming down with the camera.
That's an interesting looking rock.
Yep. A couple fishes here. Synaphobranchydial on the seafloor, as well as
Zoom in there, Dad.
Halusar in the water column.
Sure thing. Look at that.
Yeah, crazy looking rock.
Oh, you know what? It's not a synaphobranchid. Something else.
What do you think this is?
I don't know. It's a good angle to get a zoom on it.
Oh, wow.
I'm going to put
I'm not familiar with this fish. I don't know. To send a picture to our scientists ashore for confirmation. So some of the things we're looking at are like lateral line structure, fin structure, sometimes pectoral fins, head shape. That is a weird fish. It's a great imagery though. I think this is really useful.
They look almost leathery.
Yeah. I'm not sure.
Okay, can go in.
Cool fish.
Every time I think I know my fishes, I see something else that confuses me.
Then here's another question from chat. I know one of the reasons to collect these rocks is to date when the seamounts were actively erupting, but I also wonder if you have any idea of how long each hotspot might have been active. In other words, what is the range of ages you expect to see on rocks from a single seamount? 5 billion years, a million years? Is it within your sampling error or uncertainty?
Expect to see a range of a few million years, yeah. Because the plate moves at a couple centimeters a year, usually. And, convert that to million year time to when it, moves to a different area. It's part of our uncertainty. We don't expect to get
look down.
Greater resolution than a million years.
Try and keep Herc centered up here.
Okay.
Have we aged any of the rocks that we have so far, or are we still waiting to send them back to the lab?
Yeah.
We have not aged anything yet. It's a month-long process, several month-long process. Because they still have to be prepared, and then they have to be bombarded with neutrons, and then put in the mass spectrometer.
So how long does aging usually take, or trying to age a rock usually take?
Drop down a little bit more.
Okay.
Anywhere from like 2 to 10 months.
Have you aged any rocks personally?
I have not.
Will you be aging these rocks that get sent off to the lab, or do you have to go back home and live your own life?
the rocks we're picking up?
Yes.
Well, they're going to go to the repository first. And then, yeah, we'll probably request some of them to be used for dating.
Come down a little
but
more.
I won't be doing any of that. That'll be some undergraduate worker working on it.
Cheap
Ooh,
labor.
another.
Oh, yeah.
Polychaete worm.
Is that polychaete? Oh,
Yeah.
Cool.
So a lot of loose rocks here if you want to pick one up.
Yeah,
Yeah, let's grab one.
Looks good here.
You happy where Atalanta's sitting?
Yeah.
Okay.
Perfect.
Can I grab the controller?
What's that?
Can I grab the controller?
Yeah, yeah.
All right.
Pick up one of those rocks.
Yeah.
We're gonna do this one on the fly. So
Roger. Oh.
So you need to move nice and slow.
Ooh, a sea star.
Oh, yeah.
Bring the minute to the left so it's in front of the camera.
You try, like, one of these guys right here. Either of them's fine. If it's loose enough.
Nice slow movements, mate. The ROV's free flying, so if you hit, it pushes it away.
Roger.
Nope.
That one's stuck.
Okay.
I'll try
Do you
a
see
little
anything
closer.
loose?
Yeah, I'll try this one right here.
Poke a few gently with the jaws
Roger.
closed.
Are they all stuck?
I guess.
Oh yeah, that one's not moving either.
Okay, move your arm to the right a little so it's
Arm
not
to the right.
blocking so much of the camera. Okay, poke a few more. Maybe the one on the laser, see?
Yeah, that one there.
Yeah, just gently touch them. They'll move right away if they're loose.
Nope. Wow, deceiving.
Totally.
Okay.
Okay. Touch a few more.
That one, yeah, those two.
Let's check this one here. Nope.
Poke them just a little bit harder.
They definitely
Okay,
look a
should
lot
get
more
there.
precariously placed than they are.
I'm gonna try this one on the ledge here. Never mind.
Yeah, all right, let me move a little bit more.
This guy right here. Nope.
Okay, move the minute out of the camera view there. Just move it to the right. Tuck it in a little. If you move it to the bottom corner of the camera.
Yep.
I think anything
Bottom
else
corner, we'll tuck it in a
in
little
the
bit
area, or is this
more. Yeah, that's good.
the only patch?
We'll move around a little bit more here.
Okay.
You're happy with it there for now?
Yeah.
Okay.
Yeah, we can. If there's really nothing around here, we can keep moving down southways. Try again. There's no specific prescription to stay on top of the ridge or on the wall, so we can just do whatever is most interesting.
Poke that one that's sitting on top right there.
Yeah, try it. Looks pretty good.
Quick. Go quick. I'm going to lose the viz here. The one down lower.
Okay. Yeah, I see that.
Do you need a circle? You got it?
Oh, in there?
Yeah.
Oh,
Yeah.
yeah.
It's loose. Sure, grab it.
Yeah, sorry, I'm just really.
Okay, hold on.
Yep.
Probably not
Roger.
basalt, but
That one's a little,
A little thin.
yeah,
It
you
is
happy
quite
with
thin,
that back row?
yeah.
We can poke a few more here.
Okay.
Maybe off to the right.
Raj.
Okay.
Oh yeah, that one in the lasers maybe.
It's a big rock.
I don't think so. Down to the right is
Yeah.
the squarish one that's loose.
Maybe this.
Yeah, I see that.
Poke around in there a bit.
That's really not loose.
Yeah, go ahead. Just touch it.
Oh yeah.
That one's probably a good one.
Let's give it a try.
Pick that one up, see what it looks like.
It's quite angular.
Quite angular.
I think we found our specimen.
Come on.
Not yet, we haven't.
Ah.
I'm gonna mute my mic now.
Poke a few more of those to the right
This one,
there.
yeah, this one it looks loose, but
All right.
That one.
yeah, Steve, yeah, this one maybe.
Third time's the charm.
Like,
Third time's the charm.
There's a few to the right there that are, or a couple right to the left, either way.
that guy.
Yeah, this looks pretty fresh, and that's like carbonate-y stuff, right?
Yeah, definitely.
Yeah.
Okay, there we go.
Yeah, yeah, grab that.
Oh gosh.
I think you found
Okay,
several loose
you got
ones.
yeah, several loose options.
There's a
Yay,
bigger one
more
right,
options.
right in front that may be loose.
Sure, take them all. Yeah.
Touch
Okay.
that one right under the lasers. It's a piece of crust.
Yeah, it doesn't look great.
I'll go.
Touch the one right under the
Gonna go
lasers.
back for that
the
one?
big one.
This one here?
Yeah.
There's a
Angular.
thick one in the top right. That one's too big, I think. That's very big.
That's not very big, and I need ballast.
So you want this one?
I mean,
It comes with a star.
2-in-1 special.
We're really light, so we got plenty of room for
Let's do
big
it.
rocks.
all right, yeah.
Okay, where are we going with this one?
It's an E or F box.
Roger.
Wherever you can fit it. Just come up away from the wall here.
Yeah. That would be nice.
It's like
I'll open the
15
box for it.
centimeters.
Look, you can see yourself in Atalanta.
Oh yeah, that's cool. Okay. All right, it's an E.
It's an E.
It's an E.
Okay.
Roger,
Expertly
thank you.
done.
Nav, that was sample 147, correct?
That's what I wrote down.
Awesome, thank you.
Stow it.
Yeah.
Thanks for your patience, guys.
No
Great
problem,
job.
Gibson.
Awesome job.
Good
Maybe it
job.
was the 5th time the charm that time. Oh.
I think that's a theme here on the 4-8 watch. Answering that question in chat, have we ever collected samples that escaped before I reached the surface? We've collected a few that have managed to wiggle their way out of our grasps. So it is a little unfortunate when that happens, but Once we usually when we get them in the ballast box or we get them in the slurp jars, they ain't going anywhere.
Look down more. Come back up this cliff here.
Since there's not much down
just above the float
on the wall,
we can gonna come back up, Steve.
We can come back up to like the crest of the ridge.
Yeah.
Roger.
It's dusty down here. Okay, can keep chasing it.
So Brandon, what kind of flow do we see here?
Yeah.
Carbonate flow.
Anti-flow. It's pretty solid stuff. It doesn't flow.
Yeah. Yeah, I don't think it's not a flow. It's just carbonate covered in crust. Limestone. You can see a pretty pronounced layer right now. That formed when this geo was near the surface.
Answering that question in chat, did you see the cool sea urchin caption yesterday? Well, my benthic boy, we did in fact actually see the sea urchin. I even got to see it up close and personal. When we tried to dissect it, it just tried to fight back, and it did end up poking our researcher. Poor Liz.
Oh, did it really?
A few times, and they were a little upset at it, but it was really cool to see, and it actually kept its form. But they're mean, because when you poke them, they poke back.
I'm sure it was entirely intentional.
Oh, yeah, totally. the sea urchin had malicious intent.
You can move our little
stubs
closer
there.
down.
At least 1, 2, 3, 4, 6 sponges grew there at one time or another. The interesting thing is they all have similar kind of patina, suggesting similar ages, but it's very tough to tell sponge ages from their stalks.
Here and I won't be able to stay on the wall. So.
Hemicorallium is the real winner here, and some of these unbranched bamboo corals. Very successful in this terrain.
Gonna come down 5.
Oh, there's a cup coral too. Solitary scleractinian, just above us.
Zoom in there if you want.
Sure.
I must have missed a cup coral. Go wide, Tim.
Mm-hmm.
I didn't see the cup coral, Steve.
It's up and to
You're
the
looking
right.
for the cup coral? Yep. Right
Oh,
here.
now I see it. Try again on the zoom.
Oh, cool. We're gonna go tail here. Auto heading's off.
Roger. Well, then it's moving you closer now.
Yep.
This looks similar to the one we collected
full zoom.
Several dives ago.
Okay. Go wide.
Thanks. They're very fragile at these depths. Usually we don't start seeing colonial species of scleractinians until we're closer to at least 1,500 meters, probably a little bit shallower down here, but at the equatorial latitudes we start to see them at about 1,500 meters. But the oceanography is slightly different further north here, and we don't often see stony corals until maybe we're closer to, colonial stony corals until we're closer to maybe 1,000. But if we dive at Johnston, possibly in the next several days, we could very well start seeing our first colonial stony coral species, which are direct relatives of those shallow water reef-forming species. There are several known
3. Want me to come around a certain way when I try?
reef-forming species that form deep water reefs. In fact, some of the most interesting.
We came the wrong way there, and it put another
most biodiverse. Some of the most biodiverse reefs at higher latitudes are actually very shallow.
Being more than 3.
In Scandinavia, there are shallow water stony coral reefs within scuba diving depth ranges.
Here while you
made up of Lophelia, until recently called Lophelia pertusa, and then we also find in this region mostly Enelopsamia, which is yellow, and
need to be
madrepora.
right about here.
Sometimes we can also find Solanus amelia, which is found typically at higher latitudes in the north and south, but we can find it down here too. They saw it last ROV cruise up
I think I want
in international waters.
I'm pulling
one of the sites in international waters.
Really hard right now.
Back in March, April, I want to say.
What's she doing? 230?
Here's a question from Benthic Boy again. Was this seamount formed by a volcano?
Yes, Barnacle Boy, it was. I mean, sorry, Benthic Boy.
Benthic Boy. No, it's not Mermaid Man. We don't have the counterpart.
While we're waiting here, Dan, I'm gonna do some checks.
Not all seamounts are formed by volcanoes. Their lithologies almost always are, but their upliftedness, yeah, it can be tectonic in origin rather than volcanic. We were at a ridge like a couple days ago.
I can't.
That
I
was
can't do anything except go
most
forward.
likely uplifted tectonically.
I would be more
potentially recently.
It's more over here.
Closer to her.
We can pass them up front if they want.
Like I can go forward and pull it, but I won't be able to.
Volcano.
On the cliff
it's now dormant, or I shouldn't say dormant, now extinct.
Like that.
The mantle plume, which I can explain more if you want.
Herc is dictated.
The mantle plume that sort of
like the head movements.
What am I trying to say?
Sample or anything else.
The mantle plume that energized, that
there you go.
I can't think of the word.
Activated.
It fed this area with magma, with melt.
And then Benthic Boy, to answer your third question, will there be a video of the Chaunacops from yesterday? Hopefully.
Better be.
Yeah, we took a lot of highlight reels, and hopefully our editors onshore will be willing to put those in.
Yeah, most likely. That was definitely a real treat. Very kind of you, Steve. I'm a little disappointed. It's not our fault, but that Dumbo octopus was really moving yesterday, and we didn't get a really good image of it. But we'll see. Maybe there might be enough there for a highlight too. Sometimes.
So I tend to move Atalanta, not the boat.
Animals are not so photogenic.
There could even be, you could do a triple highlight, because that was within 20 minutes we saw the Chaunacops, and then the octopus, and then the big cutthroat eel, all in a row.
I don't try and work out.
That's true, we have the triples up.
If
very
I want
fishy
that to
dive.
go that way, I tell the boat to go that way.
That's rude.
Try it, see what
for
happens.
the octopus. What did Dumbo octopus eat?
Right now we need to go
I believe they're
Herc.
macroinvertivores, so that means they eat large
right now it needs to do this.
invertebrates.
Right.
You know, maybe like that worm we saw might be a prey item.
284, 285, 280, somewhere in
or
there.
potentially, they could also eat.
That's the way it needs to go. It needs to get closer to hurt.
Marine snow particles if it came down to it.
Yeah.
I don't know if they're necessarily predatory. They're.
Whatever that
I'm guessing since they were floating in the water column, they were probably.
Yeah.
This one was probably searching for prey or just feeding on material that it found in the water column. They're not active, very active predators like squids or something like that.
That's where it needs to go.
Do they travel far?
Simple as
I don't know. I would imagine they probably do some sort of vertical migration, but I don't know at what speed and at what interval. Presumably, it was going downslope because it had previously been upslope on the seamount. I don't know if they feed much above the seamount or if they stay close to the benthos. We usually find them in contact with the benthos, which is why it was unusual to see
really
it
matter
swimming down.
We just
Are cutthroat eels real eels?
I was going to answer that question.
Yes, they are eels. Yeah, true eels. Yep.
Yeah, true, I guess, not real.
Probably not I don't
I come around counterclockwise,
they
they'll bring those wraps out, so it's
have any relatives in shallow water, but most of the eels people are probably familiar with are congrids.
Oh yeah.
So I think it might be a different family, but I can double check on that.
It's a cool little fracture.
Checks are done, gauges look good.
Deep water is down to 3,700 meters. So yeah, the exclusively deep sea family.
Synaphobrachidae?
Synaphobrachidae, yeah.
Synaphinifina.
They're fairly distinctive. Yeah, we do see in deep waters, not this deep, but we do see congrids from time to time, which are in the same family as those eels that are found shallower. We can find congrids in mesophotic depths. I think in shallow water too. And then there's another family. I think the morids, like the morays. Yeah, that family.
That's a moray.
Are pelican eels Synaphobrachidae?
I don't know. I've never heard of that animal.
Gulper eel, the one that enlarges itself.
Oh yeah, I don't know. I don't think they're Synaphobranchids. I think it might be a different family. That's a very distinctive morphology.
Yeah, it does. Sure.
Sometimes you gotta stretch your legs. I encourage all of you viewers at home as well to take a little bit of a stretch break as well. It's good to keep your blood flowing and if you're sitting for long periods of time.
I feel like the moray eel is what I imagine when I think of an eel.
Yeah, probably most folks, right?
Yeah.
Or the
the electric eel called? Or whatever.
Yeah. Or the migratory eels that are, can't remember now if it's diadromous or anadromous. One of those words where they return to the Sargasso Sea in the North Atlantic for spawning, but then they will migrate up into the fresh waterways of North America and Europe.
Oh.
Yeah, so in the North Atlantic, eels have very distinctive life cycles. Highly recommend those interested in this group of fishes. They're really enigmatic. They still don't know where they spawn, but they go through these massive migrations to spawn in the North Atlantic in a place called the Sargasso Sea. And then some way, larvae and fishes find their way up to these streams, rivers and streams.
So they have no problem with fresh water?
I think they can, yeah, go between. Yep.
Oh, wow.
There's a word for that, huh?
Probably.
Trans something rather.
Trans water fish? I don't know.
So,
That's definitely not what it is.
trans water fish.
Right.
Catadromous. That's
Catadromous?
Catadromous is, yeah, where they will migrate to spawning locations.
So, Steve, here's a question from chat. Are these deep water coral able to survive in shallower water in nature or perhaps for research?
Hold on, say that again. Were they able to survive where?
Are these deep water coral able to survive in shallower water in nature or perhaps for research?
Well, so we the general answer is no. In the wild. However, there have been some instances where deep water corals have been sampled from depth, usually shallower than 1,000 meters, but sampled from depth and brought and maintained in kind of flowing seawater systems in labs. Of course, you have to keep the waters refrigerated, which is a challenge. And for every degree C you decrease the temperature, it gets more and more difficult to keep the water at temperature comfortable for these corals. So a lot of the corals that have been kept in captivity for experimental purposes are typically found between temperature ranges of 7 to 12 degrees C. We're substantially cooler here because we can maintain special conditions in the lab that would allow us to keep them for long periods of time and run experiments if needed. A lot of those experiments have largely targeted the responses of corals, deep water corals, to changing climate conditions in the ocean. So, for example, changes in pH, changes in temperature, perhaps exposure to things like oil or chemical dispersants. If they're potentially going to be in areas that might be impacted by oil spills. In addition, there's been a lot of interest lately in how mining plumes, which is basically, when you're mining or digging up the seafloor and you suspend a lot of dust and sediment, how those plumes are going to impact corals and sponges, perhaps that might be present at depths where those activities might occur. So those are kind of things we can set up experimentally.
And then also from Benthic Boy, part 4, are pelican eels and gulper eels the same thing?
According to the book I was reading, they are. It's on Nautilus. There's like deep sea fish guide from like early 2000s or something. Yeah, I called them the same thing. They also had like rockfish and hagfish and
I love
chinocops.
hagfish.
Hagfish?
Yeah, it's one of my favorite fishes.
Yeah, they're the only living relative of the conodonts, which are very important.
There's an unusual kind of reflective white blob right here. Is there any chance we can get down to look at that?
All right, Kukui, be sure to put
right
down reflective
here.
white blob.
On it.
Give it a second, give it a second.
I think it was right in this area. It might just be a patch of sediment. Yeah, this is where I was looking here. Yeah, just sediment. Okay, no need to go in there. We can come back out. However, just above this.
Is that a sea spider?
is potentially a sea spider. Yeah, I was just seeing that.
I think it is.
Yeah, can we look at this Chrysogorgia here?
Eating good.
Damn, he's
I'm curious. Yeah, I'm curious what it's doing.
You bet.
Wow, look at that.
Vibing.
That's super nice.
So there's actually there's a lot going on with these Chrysogorgia colonies. I mentioned several dives ago that these are very important for animals that might lay eggs, for example. And there's actually an egg case in this Chrysogorgia right in the middle. There's a green blob. We had hypothesized that those were actually Dumbo octopod egg cases from previous expeditions. We've dissected them open and found material that is suggestive of, some sort of cephalopod, but that work is ongoing.
You think the spider could eat it?
I don't know. I would guess the sea spider is probably most interested in the coral. It's much more low-hanging fruit. Doesn't have to work as hard. Those egg cases are coated by a very thick protective layer.
Answering that question in chat, does your watch have a name? Yes, we're Delta Dan of the benthic storms.
And there's these weird things. I've seen these elsewhere too, like these long projections. I have no idea what they are. I think maybe potentially they could be some sort of, gall or something that Chrysogorgia forms on associates that it doesn't like parasites. But this one, it looks like, so you can see the proboscis in the lower left. It looks like it's wrapped around maybe part of a polyp, or is about to be. This is a pretty obvious predation event. There's also there's a bunch of amphipods in here, like small crustaceans. These are very common in Chrysogorgia as well. It doesn't look like it has its proboscis around a polyp right now, but it could just be hanging out, waiting to take its next meal. It's a very slow process, digestion in the deep sea.
You see this?
Oh yeah, that looks like maybe a small shrimp, or a exoskeleton of a shrimp.
Interesting.
Sometimes it looks like it might have just been like an exoskeleton that the coral snatched up with tentacles. Yeah, very cool. It doesn't look like active predation's going on though. I can't even see which part of the colony would be predated. Maybe right here, like there's a lower density of polyps, so that maybe that's where the
that falls in there, is it, Dan?
Yep.
Yeah, maybe that's where the sea spider was going for.
Such cool animals.
Yeah, very nice.
So it's proboscis?
Yeah, they have a long proboscis that they use to, often they'll stick it into, their prey item. I've seen them eating sea cucumbers before, sea anemones, corals of all kinds, and then they will inject like digestive juices that will dissolve and digest the animal externally, and then it'll just slurp up kind of like the nutrient goo from that animal. It's really cool.
That paints a pretty picture.
Yeah.
Yum.
Yeah.
Let's do another 20 west.
And that feeding appendage is called a proboscis?
Proboscis, yeah.
I always thought it was pronounced proboscis.
Yeah, so did I.
All right. That's a great zoom. I think we're all set here.
Yep.
This one's
Sea
like
spider.
a nice orange color too. Last one we saw was more like a tan.
Yeah, they're variable. Yeah, darker orange, tan. Those are the common colors. Perhaps as they get bigger, they get more pale.
Have you seen any other colors aside from that? Like any other abnormalities?
Not really. I mean, I've sampled sea spiders from shallower water that are darker in color, but they're much smaller, you know, maybe like no bigger than this or so.
You can
like
start
this.
coming up.
I don't know if I believe they're also predatory.
And
come around counterclockwise here.
look straight down.
This is an interesting opportunity to talk about
don't come around
gigantism in the deep sea.
Please talk
come
about
up.
Yeah.
Oh, come up.
We find that in the deep ocean and even at higher latitudes where deep waters are often emerge, and there's a lot of mixing of deep and shallow waters going on. This is particularly apparent around Antarctica, but there's a propensity for gigantism, or animals that are normally appear small in shallow water habitats to have very large body sizes in the deep sea. It's thought that might provide like a refuge, you know, from predation or things like that. But yeah, there's a propensity for larger body sizes. So one good example is the giant isopod in the deep sea. And we see giant isopods that are the bit, you know, size of an American football. But, you know, in most other shallower habitats, they are tiny animals. Like, if you can imagine a roly-poly you might find under a log, that's an isopod from land.
Delta 1-5.
We don't find giant isopods on land. Same thing with sea spiders. In shallow water, sea spiders tend to be very small, but there's a genus of sea spider called Colossendius from Antarctica, as well as other parts of the deep sea, that is quite large. Not too much of a stretch to say it's probably like leg span is 30 centimeters or so, or it can be up to 30 centimeters.
No, I want to keep Atlanta right over.
Also, crabs are famous for gigantism.
We're going to do some
deep
tether
sea.
management here, so I need
Delta
to be
20.
do this.
the biggest crab that you've seen.
There's a on the seafloor here. I don't know. The tanner crabs from the west coast, as well as the red crabs from elsewhere, get pretty big. You know, their leg span can be 20 centimeters or 30 centimeters sometimes.
That's almost a foot, right over. For those of you Americans at home that don't know the metric system like me.
But yeah, Japanese spider crab is a huge
crazy
crab. Yeah, they get quite big. They live fairly deep as well.
Whoa.
I've always wanted to see one. They're enormous animals.
Macrocariae? Caria?
Macrochyra.
Chyra.
That is one scary looking crab.
Yeah.
Harmless.
Just a little.
Enormous.
Harmless.
Have you seen those pinchers?
Yeah, you're gonna be all right.
Alrighty then.
They're tiny.
Hey,
Steve,
Okay.
Picture the guy holding that.
We're just gonna
Yeah,
we're gonna
tiny.
come out to blue water here to do some cable tether management for a minute.
Copy that.
Struggling with the one thruster.
All right.
3 turns in the wire now.
Steve, back to you talking about, like, in regards to deep sea gigantism as well. I know you were talking a little bit about, like, polar regions, how animals get bigger there.
Yeah.
I remember learning years ago, though I can't exactly remember why, there was a relationship between, like, surface area and volume and, like, heat transfer, and how animals near the poles get a little bit bigger rather than, like, equatorial ones. Do you know much about that?
I don't know, not enough to talk about it in an informed way after waking up at 3 a.m. But I'm sure if you catch me later, I'll catch up. I'll read up on it.
Gotcha.
But yeah, I mean, we will. Part of my master's work was working in the Southern Ocean on using deep water towed cameras to assess biogeographic and habitat characteristics of seamounts and like margin areas in the western Antarctic Peninsula.
Mm-hmm.
And it's absolutely true. You do find massive sea spiders there, and, you know, enormous like sea cucumbers and things. Sea cucumbers are another good example too, because there are actually quite a large number of smaller sea cucumbers. They can also get quite big in shallow water reef environments, but you know, like solid sea cucumbers, for example, they're sedentary, so they will like attach to the seafloor, but they will feed with their feeding appendages, much like the sea cucumbers here. You know, will use those appendages to kind of move sediment to their mouths, you know, deposit feeding. There are sea cucumbers that will just use their tentacles to feed in the plankton.
Gotcha.
So yeah, definitely large animals in the peninsula.
Cool, thank you.
Cup corals also get very large in the western Antarctic Peninsula. Sometimes we find very large, there's a species of flabellum cup coral that lives in the Antarctic that gets quite big, maybe about the size of a fist or larger. Yeah. But yeah, there's a lot of really interesting Antarctic animals. Interesting thing about the Antarctic continent is that at least until recently, many crabs had not invaded Antarctica. So they, like spider crabs were absent from the Antarctic, but there was a really cool finding of several months ago. I don't know if anyone was following about the finding of Shackleton's ship in the Weddell Sea. Anyone see that?
I heard about it, yeah.
Then really quickly answer the question, when's the next photo album come out? Soon, whenever we get all of our pictures put together and stuff like that, then it'll come out. Continue, Steve.
So the crab situation was on this the wreck of the Endurance, right? It's called Endurance, is the Shackleton ship. Anyway, some intrepid scientists in the UK at the British Antarctic Survey had looked at some of the imagery that was posted by the team that went searching for that ship and found actual evidence of crabs on the shipwreck, crabs that had not previously been found in Antarctica, which was kind of mind-blowing.
It's really cool.
Yeah. How, you know, archaeology can also inform biology in some cases.
That's just another win for exploration versus research, right? Wouldn't have found it without finding the ship.
Answer the question in chat. Are you heading up? Nope. We're just getting Atalanta and Hercules untangled, just to get the tether out from around them. Steve, here's a question for you. Could this type of giganticism be responsible for the appearance of a giant squid? Or would that be too dramatic and rare of an example to be explained?
Oh, that's a possibility, yeah. I don't know about a lot of cephalopod biology specifically, but that makes sense, yeah. You find colossal squid and giant squid typically at higher latitudes and deeper. So definitely a possibility. Those are kind of like the sea monsters of today, I feel, in some ways. Like, I would love to see one, but they almost have a mythical status.
Yeah, all the cool footage that I've found so far online of colossal squid. A lot of it is just kind of like you know, B-roll from like oil tankers. Just happen to find them. I can't imagine how difficult they are to find.
Yeah,
if you actually go looking for them, you know.
There's been some success with biomimicry, using electronic like bioluminescent patterns to attract prey item.
Oh yeah.
Prey items. Yeah, you heard about that?
Yeah, is that like pulse kind of jellyfish that?
Yeah.
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 I remember seeing that.
The burglar alarm hypothesis.
Yeah, that's what it was.
Another question from Bent Boy. Do colossal squids live in Antarctica?
I'm not sure of their distribution. Something you can definitely look up, though. I don't believe they're found around here.
Have you ever seen one?
Apparently they're also found in the Mariana Trench, I think.
I wouldn't be surprised.
That's fun.
Nav, if you have a second, can you drop a target where we left the bottom? If you.
Can you guys repeat that?
Yeah, if you could find where we left the seafloor and drop a target there so we can see where we left off.
Sure.
Not that we're going to go right straight back there, but that we have at least a reference point.
Thank you. I know there was like 24 questions while I was gone, but
yeah, it was all asking for you.
Oh my God, Brandon. What is a mantle plume? Well, let me tell you, John Doe.
Let me tell you, Benthic boy. Here's what a mantle plume is. Please enlighten us, Brandon.
What is a mantle plume, Brandon?
Yeah, exactly.
That's it. That's what it is. It's a mantle plume.
That's wow. That's real descriptive there, Sherlock. Please, any more detail?
Sure. It's like a deep mantle originating plume of material, extra hot material, and that's why it's able to rise. Because when it gets hot, it's less dense, and so it rises through the mantle. And when it gets to the lithosphere, it sort of spreads out, and it's so hot that it creates melt, and that melt can travel through the crust and make us some seamounts like we see here.
Beautiful.
Do they just make seamounts, or do they also make stuff on land?
That's a good question. So
Yep.
They can make stuff become land.
How?
Because they make giant islands.
Ah.
But I don't think they can penetrate the continental crust. It's too thick.
But what about like hot spots, like those kind of mantle plumes when they like extend islands?
That's what all hotspots are, mantle plumes.
Nice.
That's sort of like part of the definition. Yeah, like Hawaii is a hotspot. Iceland is a hotspot, and it's a mid-ocean ridge.
Oh,
Really?
wow.
Yeah, it is both.
I did not know that.
Very interesting.
Oh, look, we got a 3-part question in chat. 3-part question. What scientific groups organizations do you share the samples with? Do the request samples in real time during the dive, or do requests for the samples happen in real time during the dive, and where can we expect to find the results of some of this research?
Well, so the samples are available through repositories that OET partners with. For example, the biological specimens are housed at the Museum of Comparative Zoology at Harvard University, and largely in the collections of the invertebrate zoology department, but sometimes we have other organisms. And then the geological specimens are housed at the University of Rhode Island's Graduate School of Oceanography, MGSL, which is the Marine Geological Samples Lab. And that is where both of those locations are where scientists can request them. They typically will leave the ship within a couple of days of returning to shore, and they will arrive at those locations, and then they'll be integrated by curation staff. Sometimes that process can take weeks to maybe a couple of months, depending on how many and what kind of what's required in the curation process. And then after that, they can be requested by scientists. At least, I'm not sure about MGSL, but at least MCZ has a very interesting portal where you can search the collections, including by collector, like EV Nautilus, for example, or Ocean Exploration Trust, and then you can collect those specimens. You can request those specimens from the curatorial staff there. But that is essentially the end of the specimens from the cruise-related work. The scientists that request them from there will have their own research projects, and they alone are hopefully going to publish on the results of those research questions that they're trying to answer. So oftentimes, at least in the biological world, it can take several years to get from seafloor to manuscript or published paper, and that would be in the best case scenario.
Oh, what's the worst case scenario?
There has been some studies that have suggested that many samples will often sit on museum shelves for decades, only because the taxonomic expertise might not be there, or they may be just not necessarily high priority collections. So it's important, though. I think the fact that museums house these collections for long term is really important, because they provide a snapshot of what animals were like, for example, on this seamount at this time, and they may be used for decades to come, maybe even centuries. So, while cruises and this, takes time to do these kinds of cruises, and they're very costly in some cases, the collections have a very good longevity.
That's the current one.
Oh, okay, good. Nice. To answer the question in the chat.
Back in action.
Good morning. How much longer will you be on the sea floor? So this is a total of a 16-hour dive. We dove around, or we launched around 9 o'clock last night. So that math is, hang on, 21, 22, 23, 24, 21, 22, 23, 0, 1, 2, 3, 4, 5, 6. It's been about 10 hours. So we have around another 6 or so hours until we resurface, but that is totally to be determined depending if the weather will shut us down. But
yeah, we're gonna evaluate conditions at the watch changes for the rest of the morning and see how it goes. But I think the intention is to be on deck sometime midday today in preparation for reevaluation of the conditions, and we'll go from there. If there's a weather window, then we can continue with maybe a shorter dive nearby, or potentially pack it up and do some mapping until conditions improve.
AKA, the rest of us get to catch up on sleep.
Okay, Steve, do you want to head south toward waypoint 5?
Yeah, it would be nice. Yeah, we can stay here. We don't have to go down the ridge or down the wall. Just kind of stay at this kind of slope and head south towards 5 and straight on through.
All right.
Are there any considerations, or are we good to go with the vehicles?
You're off SPO.
Yeah, I can't hear y'all.
A little challenging to orient Atalanta
Okay.
to the counterclockwise direction, but well, I'll just look down and we'll try and keep it over top of Hercules instead of having to rotate it.
Got it. Yeah, let's head on down. There's stuff to see up here on top, so we can just kind of cruise along, peek down every once in a while below the rim of the wall here.
Answer that question in chat. Is it dive, dove, or launched? It's a little bit of both, or sorry, all three. Yeah, it's when we're launching, it usually when launch means we're on deck putting into the water. Diving or dove is when we're already in the water descending down or already on the ocean floor.
And it's walking itself over right now.
What do you think about doving?
Doving is not a word, so I don't think a whole lot.
Says who?
Says me.
Webster.
Hang on.
Webster says it's not a word.
Ah, Webster.
How many 6/8 is like?
Oh, never mind. Dove as in to stun.
Roger.
So not the kind of thing that you're thinking of.
To stun. That's what I'm thinking. Like if we can afford a few more, I can come over to the right. But
that's got to be some sort of archaic or historical term.
Hang on, let me.
You know, H.P. Lovecraft was using it. When people saw Cthulhu, they were doved.
Will do. That's a name I haven't heard in a while.
Cthulhu?
Well, yeah, Lovecraft. Yeah,
understood.
Yeah.
I've been reading his short stories.
Have you read At the Mountains of Madness?
I have not.
That's one of my favorites.
I like The Color Out of Space.
Yep.
Didn't he like write those books in 3 days? Something like that?
Really?
Yeah.
Oh, I have no idea.
H.P. Lovecraft was insane.
Wow.
He's written a lot. A lot of short stories.
Yeah, he did it in a very, very short amount of time. And apparently he was like, he's not a good dude either.
He came up with some messed up crap, so.
Yeah, well.
Can't say I'm not surprised.
He's also, I think he was a rampaging racist. Like he wasn't a good dude at all.
That's a steep wall.
Raj.
That is a steep wall.
It's these kinds of environments I wish I could scuba dive in. I mean, I've been diving a fair bit around some pretty steep terrain, but this is extraordinary.
How deep do you go?
Oh, no, not crazy recreational limits.
Oh, you were talking about scuba diving.
Yeah.
Okay.
No, I always used to like doing wall dives.
Yeah.
Ooh.
Those are some
I'll
of my
try one
favorites,
day.
especially if you get a drift dive in as well.
My favorite habitat is actually the spur and groove.
Oh, spurs and grooves are awesome.
Yeah, because you can kind of like swim down in the grooves and you have like corals on either side of you.
And there's like a nice sand patch. You don't have to worry about crushing any corals with. Yeah.
And sometimes there are localized channel like currents that carry you. You don't have to move very much.
Oh, that's fun.
Yep. Spur and groove. Where were we? I think we were at Kingman Reef earlier this year, where we saw like very deep at 1,000 meters plus, like what looked like, very old spur and groove formations.
Ooh.
Probably from lower sea level stands.
Interesting. I always wanted to try to dive. I think
Roger.
like a lot of the smaller islands, like Molokai, you can see over the top when you're in an airplane, like all those spur and groove formations that look amazing.
Yeah, yeah.
Never been scuba diving. Always wanted to,
It's really fun.
I haven't
I
either.
like it. Ooh, y'all should try it.
Don't you have to get certified and all sorts of things? And
Yeah, I mean, there's also classes that are offered where it's kind of like an intro to scuba diving. So you don't really get your certification. You just go diving with a dive master if you want to try it out and you're not really completely sure you want to get certified. So they do offer those kinds of classes. It's a little bit pricey, but people seem to enjoy it.
Yeah, and if you don't want to do an open water environment, you can also do courses that are called tri-scuba, and you could just do it in a pool
Single-digit altitude and come up a little.
environment.
Oh, nice.
Yeah, those are really nice. Usually they're just one day. You can do a little bit of classwork and hop in a pool, see what it's like to breathe underwater, and you will never, ever, ever forget the first time you do it.
Yeah.
That much I'll guarantee you.
I think I was tripping out for a whole day after my first scuba dive.
you weren't there, man.
Yeah,
There's like fish and stuff. Oh my God.
was yours in the ocean, Kukui?
Yeah.
Ah, there you
It
go.
was amazing. How about yours?
In a frigid rock quarry in the middle of Minnesota.
Ooh.
With a whopping 2 feet of visibility.
Haha, yeah.
I've done a couple of quarry dives. They're actually terrifying.
Yeah, they're not my bad. I did tell people though, it's if you can dive in cold water lakes, you can really dive anywhere. So it was a good learning environment.
Yeah, quarries are a great place to learn about oceanography well, not oceanography, but like how water
affects the human body.
No, well, how water stratifies in like lakes and in the oceans. Yeah, because you can the surface water can be like 25 degrees C, or up to 60, 70 degrees F or something like that. But you hit that thermocline and it drops to below 32.
Oh, that's fun.
It's pretty striking.
Yeah, you feel it right away too.
Really?
You can hit 1 or 2 thermoclines really easily in lakes, and especially in the Great Lakes and Lake Superior, you can really quickly drop into the 50s and 40 degrees Fahrenheit.
Oh wow.
In the ocean, like where we are now, we hit our thermocline on this site at
I'm gonna come up
pretty
a few
deep.
meters.
It was a couple 100 meters. Yeah, around 125 meters it started. 200, yeah, maybe around 200 meters.
That's a very steep one. Wow.
Yeah, it doesn't get much.
But in more still water, you can get it within 30 feet.
Right. It doesn't get much better than warm ocean diving.
Yeah, come check out Hilo. You can see a reverse thermocline, which is fun.
Reverse thermocline.
Yeah, we have these freshwater springs that pop out everywhere, and so it's like super cold out on top. It's probably like still like 70 degrees or something, but when you drop below, it's a little bit warmer.
That's awesome. I didn't know that was a thing.
Yeah, that's cool.
Yeah.
Yeah, I want to try a rock quarry one time. What kind of wetsuit do you guys have to wear to dive in those conditions?
Usually for freshwater, I mean Minnesota, it's always going to be a 7mm.
Nice.
And you know, in the Pacific Ocean, where I am in California, even the most southern parts of California and San Diego, most people year round are doing 7mm.
Oh wow.
Some of the hardier people do 5mm, and of course it depends if you're doing open cell or closed cell, you know, freediving suits or just standard scuba suits.
Can we zoom in on the rock ledge here? Is that just coral bases?
Thank you, people in chat, for enlightening me on Lovecraft.
Yeah, it's just bases. Another thing, Dan, if you have time, right here, the thin stalk. A partial zoom if we can. The one right above the lasers, yeah. What do we got here? Looks like a bamboo coral. You can see one of the nodes at the base there, but yeah, unbranched bamboo coral. Okay, thank you. Is it problematic if we stay kind of right on this angle going south, or should we head up on top of the guyot more, or what's... Yeah. Right. Yeah. Okay. Yeah. You know, I think maybe we could move a little bit more on top of the guyot as we move further south, kind of keeping a southwesterly direction more so than south. Does that sound okay?
Yeah.
Let's do that. So maybe maybe aim for a waypoint like 100 meters to the west of waypoint 5.
Okay, sure.
What does that look like? Can you show me on HiPAC?
Yeah. It's about there.
Yeah, let's do that. Put a
Okay.
target there, and then let's aim for that.
Then to answer this question in the chat, do any pharmaceutical...
Yeah.
You feel a strong current, yeah? Huh, okay. That's good to know.
Answering that question in chat, do any pharmaceutical companies ask the expedition to look for certain corals, sponges, or fish to see, to study and to see if they develop new medicine? On this expedition, no, because we are in a wildlife preserve and a sanctuary protected by the U.S. government. We are not allowed to take those kind of stuff aside from the stuff that we're trying to study.
Yeah, no commercial purposes.
Yes.
Yeah.
And I don't believe, I'm not entirely sure that Nautilus has actually teamed up with pharmaceutical companies before. I could be wrong in that. Steve, do you know?
I don't believe that's the case,
Yeah.
but I would invite those who are interested to take a look at a cruise we did a few years ago. NA128, I think. Southern California borderlands. There was a team out there that was doing some really cool work with natural products chemistry. So I think it was an A128, which you can find on the website.
Yeah, at nautiluslab.org.
There was exposed limestone there.
Ooh, exposed limestone. Limestone.
Too strong.
Right. Huh. That's interesting because it's directly contradictory to what the currents were suggested for this site.
Oh, really?
Yeah. Yeah. Well, that's interesting. Still, yeah, interesting observation. We were expecting currents mostly out of the west-northwest.
Looks like it's, yeah.
I think it is coming out of the northwest.
Oh,
Yeah,
it
it
is.
is.
Oh, I thought it was coming over the geo, like out of the west-southwest.
Does that look like exposed limestone? That like brown?
Yep.
The tan thing? Can't tell if that's a coral.
Okay, well then that makes sense then.
I think it's a rock. Snooping.
Sure. Yeah, I think it's a... Yeah, I think it would be better this course that we're on towards 5.1. I think that's a good angle, and then we'll kind of evaluate what direction we want to go to when we reach 5.1. Probably still head maybe south, slightly south and west from 5.1.
Copy that.
Yeah. We'll decide when we get there. So what did the exposed carbonate look like? I didn't catch that.
It looked like a rectangular, like light brown. I wasn't sure if it was coral, sponge, but it didn't look like sponge. It didn't have like holes in it or anything.
Yeah.
But it was the only one, so it'd be kind of weird.
I love when we're shallower, where there's not so much crust, and sometimes if you get fresh breaks, you can actually see coral embedded in the rock. Yeah.
Oh, cool.
And we collected one
Delta for you,
a couple of years ago that from
Delta 1.5 is a good target.
Well, maybe it might have even been last year, but it was intended to be a rock collection, but we cut it open and it was a big chunk of coral.
Not fully lithified yet?
Yeah. It was quite deep too.
So, Steve, here's a question for you. For the coral and sponges, how long does it take for them to create a base attachment to the rock and other structures, as by the video streams and bases seem to be very strong looking?
Understood.
You bet. De-stress. Nice and relaxing.
Beautiful.
Beautiful.
Yeah, wow, this is a really fleshy one. Bamboo coral node branching. It looks like in most cases there's at least one, maybe two branches arising from each node. Really fleshy. Well, since we haven't got much going on, do you think we could sample a piece of this?
Sure. Yeah, I'll stop her up.
Bamboo corals are a little bit of an enigma. I mentioned we use a clade system for classifying bamboo corals, and very few bamboo corals are properly named with a genus and species identifier, and even still, it's somewhat unreliable. For example, what we call something in the genus Isidella, which is an unbranched typically bamboo. Whip-like morphology can arise at multiple places across the bamboo coral family tree. So,
sorry, Steve.
Yeah.
Any particular spot you want?
That's
Circle what you want.
That's a great branch. Yeah, like a branch like that.
Roger.
Or yeah. Or even the one lower, if this is better.
You want the whole branch, do you?
Yeah, like 10 centimeters or so. Maybe a few branch points in there.
Yeah. Roger.
Generally, our sampling is very minimal invasiveness. We try not to take more of the coral than is needed in the case of colonial animals, but with the idea that these collections will be used for decades or centuries, it's important to get enough for voucher specimens.
Turn the bender off for me.
Yep. Off.
Right.
And in addition, this work has a more timely goal objective to support some of the eDNA sequencing we're going to be doing over the next several months. eDNA and is kind of like making a puzzle, putting a puzzle together. eDNA might show you, the outline of the community or the outline of the puzzle.
Zoom in a bit more there if you want, Dan.
Hey,
and the puzzle shapes, but
Thanks.
to see the actual picture, we need information about the organisms that live here. Oh, NA.
That kind of be about right there.
That looks great, yeah.
Swing and a miss. We can go. Yeah, perfect. Thank you.
We can go in the starboard box with this.
Yeah.
Starboard box?
We can go, I feel like we can go either lambda or omega.
Either where?
Yeah.
Yep.
There's only a rock and a narella in
Okay.
lambda.
Actually, yeah, we could go in the forward box.
Forward
on
box?
Lambda, yep.
Roger that.
There's no visible associates on this, so no real chance of getting mixed up here.
Just closing the starboard box. All right. Ready to come out when you are?
Yep, go ahead. Left side, right?
Left side, yeah. This is a morphology we haven't yet collected on this cruise, and bamboo corals are very poorly represented in the collections from past explorations of this area.
That is correct.
Very nice.
Beautiful.
Gentle touch.
Answering that question in chat, is there Nautilus merch available, shirts, hats, etc.? Yes, there is. I believe it's just for the crew though. We all have our little, we got a little hat, a shirt, and a travel mug.
Dan, while we're putting our toys
cool.
away.
You bet.
Don't forget about the patch.
Oh, and the patch,
and
we
the
also
sticker.
we got some patches and stickers that were also applied to us. They're pretty cool.
It's a good close-up here of the polyps. When they're closed, they have a slightly different morphology that might tell us something about their colony's identity. It looks like the polyps here are
on bubble. You're aware that the starboard box doesn't seal, right?
only extending out
Right now?
two sides of the branch, so
Yeah,
It keeps
in
them
this case the
sliding
left
out a
and
little
right
bit.
side.
Yeah, so
Okay.
Some colonies have polyps all the way around the branch, some will have only one row of polyps, so this is very useful information. All right.
Okay, you can go in. Why?
Thank you, person in chat,
Let me
for
catch
blessing
up to
me
you here.
when I sneezed. I appreciate you.
Somebody said bless you?
Yeah.
Was it Benthic Boy?
It was not Benthic Boy, but Benthic Boy does have another question.
Oh.
What samples do you have so far?
Let's see. We have 4 rocks. Make that 5 rocks. We have a narella, a bamboo coral, another coral, a blob-looking thing, and
my personal favorite.
The purple blob.
Purple, yeah.
I think that might be my favorite too.
Well, not purple. There already
DC
is a purple
grab
orb,
back
so
on.
it would be
Yes,
purple,
please.
purple lump, I think we call
Purple
it.
lump, yeah.
Yep.
Purple lump. And the tunicate thing that looks like a brachial pod.
I'm a snuffleumpagus.
Give myself a little bit more.
This is just the dive of oddities, which is good. Sometimes you learn
more from those.
You learn interesting things.
Then here's a question for the ROV pilots. If it was allowed, is Herc capable of doing things like barrel rolls and loop-de-loops?
Whoa.
Dan's already stressed out.
Yeah, I'm not on SPL at the moment. Sorry, I can't.
He didn't hear that on SPL, but they're asking Dan if Hercules can do barrel rolls and loops if it was allowed. I'm going to take a stab at it and say likely not.
Yeah, the vehicles have a foam pack on top, which basically means that they only float in one direction, one orientation.
Yeah, I think we're ready for a move.
It would take a lot of thruster power and a pretty big moment arm to flip that thing.
A pretty big what?
So if you put your thruster...
And it really doesn't make much sense.
No, but
why you would.
Yeah.
You need to generate a moment large enough to flip the heavy bottom on top, which means a long arm and then a force vector far away from it.
Huge Faerid. I believe that's a Faerid. This is a live individual of a lot of this dead sponge that we've been seeing.
Also an
falling
indication
down
of
slope.
the current there, Steve.
Yeah,
Yeah. It's ripping.
Also, it indicates too that it's fairly consistent because the sponge has been growing in that morphology has been growing in that manner for some time.
And it will grow in the direction of the current?
Well, they grow in the direction of least resistance, right? They don't want to be, you know, fighting. Yeah.
I know how they feel. Sorry, I was really struggling there for a while, so we're. I couldn't figure out what was
this might be some of the strongest currents we've had.
I think so, yeah. Just
so
took
far
me a while to realize it.
Yep.
What's going on over there?
I don't know how to spell Faerid, and it's coming up with a different with everything. I'm trying to look on what it is.
We have 3 things going on. We have 1 thruster on.
Steve, how do you spell Faerid?
Well, let me look up that ID and make sure that it's consistent with what I'm thinking here. There's another option, but let me take a look.
And we have weather and the boat's challenged, so.
It's like Faerridae. Has the
all on a very steep cliff at one point.
Yeah, and we're trying to stay on the cliff.
There's also a potential that could be a Tritopleura, which is a different group.
Good luck spelling that one.
Yeah, I think this one is actually Tritopleura, slightly different, which is actually in a different family, called the Untitled.
And how do you spell that?
Uncinateridae. Genus name Tritopleura. Tretopleura.
If for some reason Atlanta creeps all the way over, puts another one in, I'll let you know.
Pleura.
Yeah, I would just.
Pleura.
Sorry it took me so long to figure out what was going on. What's happening is there's a really strong current, and you were changing your head to try and do what we
We
normally
were
do,
seeing
keep Hercules
Faerids
in
a bit
view,
earlier,
and then
but
the current would get
right
us,
here it's mostly
take us
Tritopleura.
around. So the way we were moving, it happened to just every time you
Right, it was too much.
every time you bring your head out of the current, it's going to spin Atalanta around because of the drag on the tether.
Yeah, right.
So if we just keep the tether downstream, or you keep, looking
Then it
into
should
the
be all
current
right.
there, yeah, then it shouldn't spin anymore.
Gotcha.
It's just tragic. It took me 3 turns to figure that out.
There was one Faerid that was collected at 2,000 meters on the east ridge of this seamount we're on right now. That was a Faerid, but it looks distinct from this Tritopleura here. I don't know how widely collected Tritopleura is, so that's something I'm going to have to look up for future dives, because we've seen it at several sites so far, but I don't know if we've resolved a good ID. The NOAA Animal ID Guide is a great resource. NOAA Benthic Animal ID Guide is a great resource. However, it's no longer being updated. And if folks are interested, there's a new guide through the DARC lab, D-A-R-C lab at Manoa, who is updating a lot of observations that they've been making from previous hurl dives over the years with submersibles, ROVs. So I think that's currently being constructed and or updated.
And then,
oh, wow.
Benthic Boy, to answer your question, what is the purple blob sample? We're not entirely sure. Some people in chat say it was a sea slug. Yeah.
No, yeah, it was. Yeah, I don't think it was. It was probably some sort of. It wasn't even gelatinous. It was probably some sort of foraminiferan. But it was interesting. We'll see what the construction of that shell-like material is.
And we'll learn that in the next 10 or so hours. Oh, wow, that's a long one. Hang on.
So it looks like we're going to leave the edge of the geode here and come up on top for a while. Is that the plan?
Yeah, that's right.
To the person who has that really, really sweet message that they just put in chat,
I'll take the bubble
Rita,
for some checks.
unfortunately, Liz and Hoku are currently not on watch, but I'm sure if you were to wait until they do come on watch, I believe they are after us. So if you'd like to wait another hour or 45 minutes or so, I'd be sure to re-ask that question, and I'm sure they'd be happy to answer it.
I can come up.
So we'll maintain your west heading there, but I'll just come out in front of you a little bit so you're not looking straight down. Seems like you're spinning around there.
Yeah.
What's happening?
Here's another question.
Is that me pulling you around when I come?
It's straightening itself out now.
Roger.
Another question from
Really?
Tony.
So I try and come forward and it pulls you around.
Yeah.
What's something you wish you could sample, but you can right now? Yeah.
So that's me coming forward too fast, and it's just the drag of the tether singing it around.
Generally, I know that our scientists ashore as well, across multiple different exploration platforms are always very interested in sampling mobile animals like fishes in situ, but there's no easy way to do that using an ROV, at least not yet.
Okay, you can look up just a little bit too.
So yeah, that's something we can't get a handle on, because I know there have been many animals, many fish species that we've seen on the seafloor that either haven't been recovered using trawls or survey type things, or they look very different on the seafloor in situ versus shallower. A lot of animals, especially fishes, when they go shallower, they experience barotrauma and they get distorted, distended.
Yeah.
Fishes are always of interest, but getting them to the surface intact
again.
is always
I'll
challenging.
come back under you.
The other thing is gelatinous plankton. A lot of gelatinous organisms will not tolerate the change in temperature, moving from deep to shallow, especially gelatinous plankton in the midwater.
We didn't catch it. It's going to kill me. I'm going to hear about that later.
And then Tony, to answer your second question, should there be a shanty about the Nautilus and the ROVs? Probably not. I don't know if someone in chat would like to make one, I'm sure we would love to.
Yeah, so it's sailing out.
There is a Nautilus album
behind.
There's a Nautilus album?
Coming out of
That's all I'm going to say about that.
That's okay. Well, there you go. There's some homework for home. Find the Nautilus album.
Steve does the hip-hop portions of that album.
Making
no,
a giant
it predates me.
Behind us in the breeze, in the current.
Great.
It is effectively shorter. So
Steve,
if I shorten
here's
up
another
one
question
side,
for you
the
from a little while ago.
And because we only have the one thruster.
How long does it usually take for the corals and sponges to build their anchor points? As these seem to be the only thing left after the sea, after they die.
Yeah, I mean,
it's possible there's no thrusters and it's just the current holding it.
Ideally,
and all we're seeing is a command.
Ideally, they continue to calcify as they continue to grow. So as they continue to grow upwards and branch out, they continue to calcify their base. So it's obviously the part that's been growing the longest, so it's probably going to be the thickest part of the skeleton.
Squiggly thing in the bottom right there. I don't know what that is.
In the sediment there?
Yeah.
Chrysogorgia.
Yeah, that's a branch of a sponge.
Oh.
Sponge?
Oh.
That sponge.
Yeah, that thruster's.
So Steve, this is more of a personal question.
Pretty much, that's like 3/4 of it.
The show
command.
SpongeBob SquarePants.
So
what kind of sponge would SpongeBob be?
Not working.
I don't know. I think that may be a.
Fully speeding that one.
I don't know if I'm qualified to answer that.
Yeah, what is that?
We need those sponge expert scientists.
Really crazy because it's turned off.
Hang on.
So.
I think SpongeBob is a sponge of the domestic variety.
Yeah, honestly.
the synthetic variety. That's the hypothesis I've always held, but not a question I've investigated.
What is that?
That's an anthomastus.
My eyes are not connecting to my brain.
See.
Oh, it says it's
my eyes are just blurry too.
Apelesnia fisturalis? Anyway.
It looks like a real sponge. It's really bright yellow.
Hang on, let me.
Fisturalis.
Yeah.
Yes, this.
So that was when I.
Yeah, I had always thought that SpongeBob was an allegory for marine debris.
To the east. But so what I really didn't get.
Square pants.
You would expect.
What kind of star is Patrick?
If there's that much current.
I think it's a slime star.
Atlanta to be like way back here.
No way.
So the.
Yeah.
There was an interesting.
Yeah. So Atlanta should be here, and the currents.
Interesting image of.
I think it's probably the motion of the ship that keeps it a bit north, but otherwise,
SpongeBob Patrick,
the West Ocean's coming
star
from
that was taken by Okeanos Explorer recently, and within the past couple of years or so, I think. It made the rounds recently again earlier this year for some reason. I'm not sure what it was all about. But I think the photo was from last year, and the New England seamounts or corner-rise seamounts.
Look straight down again if you're not already. I'll try to get back out ahead of you
sponge
just a little bit. So
star side by side.
Aww. Was it a predatory star? I think Patrick's a predatory star.
What was
don't ask questions you don't want the answer.
Oh, no.
So we were coming up and down from the edge.
The world is not what it always is like in the cartoons, unfortunately.
It's just
so then we had control.
Patrick is a Chondraster starfish?
The current would blast us.
Chondraster?
What does it look like?
Great question. What does it look like? It looks like Patrick.
Kind of looks like a slime star.
Come on a little bit,
Because
they don't need sponges, right?
Sure.